My name is Kaori Yamada. I am an assistant professor at the Department of Pharmacology. We are studying the mechanisms of the trafficking of signaling molecules to regulate the angiogenesis in cancer and eye diseases. As we are studying angiogenesis, let me start from what is angiogenesis. The circulatory system is important to supply nutrient, oxygen, and hormone to the cells, and remove carbon dioxide from the cells to maintain homeostasis of the body. During the developmental stage, new blood vessels form from pre-existing vessels. It is called angiogenesis. The adult body has fully developed vasculature, so angiogenesis is limited only when it is really needed, such as wound healing. However, if the balance is broken in the disease condition, massive angiogenesis is induced, for example, in cancer and blinding eye diseases. Cancer cells need oxygen and nutrients to grow. They secrete a growth factor called VEGF, vascular and cellular growth factor, to recruit blood vessels, which supply more nutrients and oxygen. Other examples of angiogenesis-related diseases are blinding eye diseases, such as wet AMD and diabetic retinopathy. Wet AMD is a leading cause of vision loss for all the patients in the United States. The hallmark of wet AMD is an upregulation of VEGF, which induces neovascularization from choroidal vessels. The newly formed vessels are very leaky. Infiltrated blood cells and plasma induce inflammation and damage to the retina, and ultimately cause vision loss. Both diseases are caused by VEGF. So blocking VEGF by its antibody was thought to be a promising therapy. However, some patients are not responding. Repeated treatment decreases the effectiveness of anti-VEGF drugs. Then the alternative strategy is switching the drugs or combination therapy with the other drugs, which has different mechanisms of action. This is our working model. To develop the new strategy, first we need to know what's going on in the cells. Endothelial cells lining blood vessels sense the gradient of VEGF, migrate toward VEGF. The first test cell becomes a tip cell to lead the group of migrating cells toward VEGF. The followers are stag cells. To sense VEGF from the environment, the receptor VEGFR2 needs to be exposed to the cell surface. However, VEGFR2 is frequently internalized and moving between intracellular membrane compartment and cell surface. We found one of the kinesins, KIF13B, transport VEGFR2 to the cell surface, and it is required for angiogenesis. So, to inhibit massive angiogenesis in the disease condition, current therapies are blocking VEGF. Our strategy is inhibiting VEGFR2 trafficking to the cell surface to prevent the response to too much VEGF. Here is the data showing the role of KIF13B. We stained VEGFR2 on the cell surface. Before VEGF stimulation, VEGFR2 was localized on the cell surface. After stimulation with VEGF, VEGFR2 was internalized and disappeared from the cell surface, and appeared again on the surface later. However, when we knock down KIF13B from the cells, VEGFR2 could not come back to the surface. KIF13B is a kinesin family molecular motor in the cells. Let me explain the basics. The cell has cytoskeletons, such as microtubules and actin filaments, to form the shape of the cells. Molecular motors like kinesins, dyneins, and myosins move on the cytoskeleton to transport molecules in the cells. Microtubules are like highways, and actin filaments are like local road. To transport molecules for the long distance, kinesins and dyneins do their jobs. Kinesins move to the plus end of the microtubules, which locates at the periphery of the cells. So it makes sense that kinesins contribute to the transport of the molecules to the cell surface. What is the function of the molecular motors? They transport the molecules to the certain area, such as the leading edge of the migrating cells. Accumulated molecules at the leading edge activate local signaling to induce cytoskeletal rearrangement, changing cell shapes, migration, and ultimately mediate angiogenesis in the case of endocellular cells. Based on the findings, we have three big projects in our lab. 1. To study the mechanisms of regulating KIF13B-mediated VEGFR2 trafficking in angiogenesis. 2. To study the role of VEGFR2 trafficking in disease progression. 3. To develop our strategy for the drug market. For project number 3, we have already shown the efficacy of our peptide in mouse models of cancer and wet AMD. Here is the data of the cancer model. We developed the small peptide called KI, which disrupts the interaction between KIF13B and VEGFR2. Treatment of tumor-bearing mice with KI significantly inhibits the tumor growth and reduce the number of blood vessels in the tumor tissue, as shown in the graph and immunohistochemistry. Developing a new therapeutic option is beneficial for human patients. If you are interested, you are welcome to try for further drug development. But as an academic basic research laboratory, I would recommend you to study the other two projects. For the first option, we know that KIF13B is important to transport VEGFR2 to the cell surface, but we do not know from where it starts transporting and to where it goes. I know it is the cell surface, but is it apical area or basal side or lateral side? To accumulate more VEGFR2 to the direction of the migration, they need to know where VEGF comes from. The molecule originally on the area is activated and sends the signal to the cells to recruit more VEGFR2. But how? By protein-protein interaction, mediating the cytoskeletal rearrangement, activation of other molecules by phosphorylation, it is not known yet. But we have all tools to study the mechanisms, such as knockdown of the proteins, inhibition of the interaction, tracking the protein by live cell imaging in 3D culture system of angiogenic cells. For the second option, we use mouse models of the diseases. You can choose either a cancer model or a wet AMD model. For both diseases, angiogenesis and vascular leakage are critical factors. In the case of wet AMD, the vascular leakage is known to happen before angiogenesis. In response to VEGF, the tip cell is selected. The cell loosens the junction with other cells, remodels the matrix by secreting the proteases. and the pericytes are detached so that the tip cell can migrate. But where VEGFR2 is localized and how VEGFR2 trafficking can regulate these events are not known. Using the disease models of mice, knockout mice, and inhibitors, we can study the precise roles of VEGFR2 trafficking in the vascular leakage, which is at the beginning of the wet AMD and which is critical for the metastatic state in cancer. About mentoring, I will work with you and show you how to do each experiment at the beginning. We have two undergraduate students who did not have any research experiences before, but they learned the research techniques and they are conducting a research project by themselves with some troubleshooting with me. They are really excellent, but I'm sure you can do that too. Also, we have a lab manager and a postdoc. They will help you too. Finally, our lab has continuous financial support, and currently we have RO1 from NIH. So do not worry about it. If you want to try some experiments, we will support you. If you are interested in joining our team, please feel free to contact me. Our lab locates on the fourth floor of Comal B. Check out our website to see more information. Thank you.